So in the second part of the lecture about G protein coupled receptors, we are actually going to be talking only about G protein coupled receptors, about their structure and function. Again, these are the receptors that can bind different neurotransmitters, and we will talk how is this binding affecting the changes. Why should you care? There are many genes actually that code for G protein coupled receptors. If you remember from the previous lecture, it's going to be your adrenergic receptors, all of your dopamine receptors, most of your serotonin receptors, your muscarinic cholinergic receptors and metabotropic glutamate receptors, GABA B receptors, purinergic, opioid, cannabinoid, and very importantly, these are the receptors through which you're going to be detecting light. So your opsins, so signaling from the vision, is through G protein coupled receptor. Your olfactory receptors are G protein coupled receptors, as well as your gustatory receptors. So pretty much they're there to help you see, smell, and taste, in addition to feel satisfied by all of those three things. So what's their molecular structure? They are all 7 transmembrane domain proteins, which means again they have 7 transmembrane domains, 1, 2, 3, 4, 5, 6, 7. What is an additional feature of them is that they have extracellular N terminus, they have intracellular C terminus, so that's kind of specific for all of them. This structure makes for 3 different intracellular loops. Intracellular loop 1 between first and second transmembrane domain, 2 between third and fourth, and intracellular loop 3 between 5 and 6 intracellular domain, and then there are 3 extracellular loops. Those intracellular loops are very important in addition to C terminal because they are the sites where these G protein are bound to G protein coupled receptors. So most of the binding of the G protein alpha subunit occurs at the intracellular loop 3, and this is actually the loop that is most diverse between different G protein coupled receptors. It can go from like 20 something amino acids to 100 or something, 200 amino acids. So the size really varies from one to another protein. The C terminus of the G protein coupled receptors is very important for its modulation. This is where different kinases are going to phosphorylate it and then affect whether G protein coupled receptors can bind G proteins or whether they're going to be internalized. For example, protein kinase A that we briefly mentioned in previous lecture and protein kinase C, and we're going to be talking much more about these kinases, are both going to phosphorylate at the C terminus, and as you will see later on, that's going to prevent binding of G proteins to G protein coupled receptors. At similar space, but at different residues, is where G protein coupled kinases, GRKs or GERKs, are going to bind and phosphorylate. Now, this phosphorylation is not going to prevent binding of G proteins, but it's going to lead to the binding of beta-arrestins that you will soon see can then lead to, it's going to prevent binding of G proteins, but it can also lead to endocytosis of these receptors. So those are very important sites that lead to the modulation. So again, intracellular loop 3 and intracellular loop 2, but most intracellular loop 3 is where the G protein coupled receptor bind to the G proteins. Where do they bind to neurotransmitters? So there are 2 different spots where they bind. Here is presented actually for beta-adrenergic receptors. They bind, the binding sites are determined by the amino acids that are present in these transmembrane domains. So these transmembrane domains form this ring-like structure that goes through the membrane. And for beta, for norepinephrine, this binding site is going to be, and for dopamine is going to be inside, within this actually flyer. You can see it's below the plasma membrane, so 11 angstroms is where they bind. So there are different amino acids by now. We know that help in binding neurotransmitter in this pocket. And obviously, when you mutate each of them, you're going to decrease significantly affinity, or you're going to prevent the binding. So that's how these sites were isolated. In addition to all of this, there is also disulfide bridge between transmembrane domain 3 and 4 that's important for its structure. Again, when neurotransmitter binds in this pocket, it's going to lead to conformational change, which is then going to lead to activation of G proteins, which is presented on this slide here. So the idea is that ligand binding, so if this is your inactive G protein receptor, coupled receptor, then we have neurotransmitter binding. It's going to lead to the conformational change in it, which will then lead to the activation of G proteins. It's going to also increase their affinity for G proteins and allow for their activation. You do not need to know the details of this, but you can just really appreciate from either side perspective here that the distances between these different transmembrane domain change when neurotransmitter is bound, which allow again increased affinity for G protein to bind to G protein coupled receptors, and also from the intracellular perspective, again, these are your loop 5, that becomes much closer to loop 3, and again, this increases their affinity to G proteins. And that's the major way that G protein coupled receptors work. So G protein coupled receptors are going to bind G proteins in their inactive form. G proteins, when they bind GDP, are in an active form. Binding of neurotransmitter to G protein coupled receptor is going to lead to exchange of this GDP bound with the GDP. G proteins, when they have GDP bound to them, are active, and then they can lead to activation of primary effectors. This is on purpose shown as a clock because that also tells you that this process will be inactivated by itself, and we're going to talk more about it when we come to talk about G proteins, but it has internal clock that's going to release this GDP and bind GDP back. But what you should remember from this slide is that the binding of neurotransmitter for G protein coupled receptors leads to their conformational change that then leads to the activation of G proteins by exchanging GDP that's bound to the inactive G proteins to GDP, which leads to their activation. What effect this is going to have downstream really depends on which type of G protein is bound to G protein coupled receptor and what kind of effector enzymes are expressed by that cell. So we briefly talked about different systems. So we can have GS bound to this beta-adrenergic receptor, which is then going to lead to activation of adenyl cyclase and CMP production. But you may have type 1 muscarinic receptor, which will have GQ, and again phospholipase, which we talked will lead to increase in IP3 and deacylglycerol and different downstream effect. Or we can have inhibitory GI, which will then beta-gamma from it is going to lead to the opening of GERK channels. It's important to again to remind you from that amplification part that activated G protein coupled receptors, so each of these can couple to many G protein molecules because this is a transient activation, so they're going to activate as many of them, which can then lead to their downstream affinity. And this is called collision coupling, the fact that each of these receptors can lead to activation of many G protein, downstream G proteins. How is response terminated at G protein coupled receptor levels? When simply as that there is dissociation of neurotransmitter from the receptor, so then it dissociates and it's clears through diffusion or through the uptake. That's one way. The other two ways is that I told you about this phosphorylation by protein kinase A and protein kinase C, which are activated through again downstream signaling. This will prevent G protein binding to it, so it cannot couple to many more G proteins, and therefore it's not active. Or we can have GERK kinase, which will phosphorylate this certain muscidisplide and lead to the binding of beta-arrestin. Now, beta-arrestin binding it's the way they're desensitized. It will prevent binding of G proteins to G protein coupled receptor, but in addition to preventing of binding of G proteins to it, it can lead to internalization through clustrin-coated vesicles, which will then effectively decrease the amount of receptor that's present, and therefore your cell will be desensitized to this agonist. Once the beta G protein coupled receptor is endocytosed, there are two ways it can go. It can either be dephosphorylated and recycled, or it can be degraded. It's another way. And to be honest, it's very little known which way it will go, whether it's going to be endocytosed and again recycled or is going to be lysed. It's still not clear, but signaling is regulating this. So what I would want you to think at home now about these problems, what we have just learned about two different domains of G protein coupled receptors. So we talked that we have this intracellular loop 3 that's very important for the binding of G proteins, but we also have this region between different transmembrane domains that are required for neurotransmitter binding. So there are two very important domains. I want you to think about these several problems. So how would you use molecular cloning? You have these two different receptors, alpha-2 adrenergic receptor and beta-2 adrenergic receptor. For alpha-2 adrenergic receptor, it's activated by alpha agonist, and their downstream effect of adenyl cyclase, which is your readout, is to inhibit it because these are bound to inhibitor GIs. The other protein or gene that you're playing with is for beta-2 adrenergic receptor, so it's binding B agonist. And then downstream of it binds GS proteins, which S stands for stimulatory, it activates adenyl cyclase. So your output measure is going to be level of CMP, which in this case is going to be, because this one inhibits, it's going to be decreased, in this case be activated. But I would like you to design a chimeric protein that will bind beta agonist, but activate GI, or bind alpha agonist and activate GS. So where would you cut? What tools would you use to make these proteins?